All right, so let's think about this. We've made it through, oh, what week is it in the class now? Week 7. So we've made it through the first 6 weeks of the class, and we never actually looked at DNA up close, which is pretty strange in a genetics class. So the reason why that is because obviously we did transmission genetics first. We did a little cell biology, then we did transmission genetics, then cytogenetics, and now we're finally up to molecular genetics. And now we can look at the molecule, the DNA molecule, as well as the molecular aspects of genetics. So let's begin. Okay, so I guess the first thing to keep in mind is that despite the tremendous diversity that you see out there in life, coding instructions for all living organisms are written in nucleic acids. And this, was not a given. It's given now. Nowadays we think of DNA as obviously this is how we encode information, and that's because we know that now. But that was certainly not the case. Even just while there are people still alive, my parents, for example, still alive today, and they were born into a world where we did not know DNA was how we transmit our genetic information. So the idea that genes are made up of nucleic acids was not accepted. So the idea that units of heredity were made up of nucleic acids was not accepted until after 1950. So, like I said, there are many people today that were born into a world that are still alive today that were born into a world in which we did not know that we pass on our genetic information using nucleic acids, DNA, RNA, etc. So, even before, here's the thing though, even before we knew anything about nucleic acids, biologists recognized that genetic material had to do certain things, how we pass on our traits. Now, you might recall from earlier in the course, I told you that, let's be realistic, even before we had a written language, like I'm talking prehistory, right? The earliest human beings still knew that if they had a child with somebody, that child would be a little bit like them, a little bit like the other person, and so you're passing on your traits in some form. And so the idea of trying to explain how we pass on our traits has been around throughout the history of humankind. And you might recall we talked about a number of different scenarios where people came up with different ideas. I mentioned the homunculus and the preformationism long, long ago in the very first, I think maybe the first lecture. But basically, that has been around for a very long time. And so, with the advent of science, and obviously we're fast forwarding quite a bit from, prehistory to, let's say 1800s, 1900s. With scientific analysis and whatnot, biologists recognized that genetic material had to contain complex information in a stable fashion because that information had to be passed on from generation to generation to generation to generation. It was stable. They also realized that whatever this genetic information is, it had to be able to be replicated faithfully and with high fidelity because if you make mistakes, then things change. And for the most part, we always tend to focus on mutations and changes with genetics because that interests us and that leads to different outcomes, perhaps diseases, basically a squeaky wheel that's going to get all the attention. But most of the time, if you think about it, we pass on our traits faithfully and they aren't changed, right? Like, I mean, you could limit yourself to just what you could see and you could say like eye color, hair color, skin color, whatever it may be. And while there might be some minor variation here and there, there's a tremendous amount of repetition as well. And so genetic material had to be replicated faithfully so it could be passed on to offspring with high fidelity so that there weren't as many changes. We also know that, or knew that whatever the genetic material was, it had to encode the phenotype. Obviously, it had to be complex because, well, you can see right there, it had to be complex. It had to contain complex information. So the thought was that it had to also be a complex molecule. And so, although our understanding of DNA encoding genetic information is, and I'm serious when I say recent, I really mean recent. We're talking like the 1950s, right? And you might not think that's recent because, wow, that's like 70 years ago. But don't forget, there's still people alive today that are, certainly, older than 70 years. And then, more importantly, we're talking about the entire history of humankind. And we've only known that entire history. We all knew that there was some way that we were passing on our traits, but we didn't know how. And then throughout the entire prehistory, throughout the entire the history of humankind on this planet, It wasn't until the 1950s till the present, so the last 70 years, that we actually knew it was DNA. And so that is the blink of an eye compared to how long we have been on this planet. And so it is recent. Our understanding of DNA encodes genetic information is very recent. And that's why there's been this explosion in genetics, biology, biotechnology, biochemistry, molecular biology in the last few decades, because on top of it being relatively new, then on top of that, you layer on the advances made with computing power and our technology, and the our understanding and manipulation of that genetic material has increased dramatically in recent years. Okay, so how do we know DNA carries genetic information? For a long time, we didn't. And so here's the story. Here's how we got to the point of realizing that our genetic information is carried on deoxyribonucleic acid and not proteins. So many people thought it was protein, as you'll hear me say. We'll get to that in a moment. But the story begins with Johann Friedrich Miescher. And so this is 1869. I am not going to ask that you remember the years, but you should be able to relate or be able to match a last name to their contribution in this, deciphering, if you will, of how we encode our genetic material. What is our genetic material, I should say. Okay, so Miescher, so 1869. You know what a lot of people had? They had a lot of infections. And so sterile technique was not nearly where it is today, and so there was no sterile technique actually, and so Miescher was studying pus, right? And so while it sounds gross, let's think about it. Back then, when you got a cut, you would get infected, and then the pus was viewed as a good thing because it would be fighting off the infection. It is, I mean, it's composed mainly of your white blood cells and your immune system fighting off an infection. And so pus was thought to be have these healing properties, and it may not be a perfect heal, but it's healing you. And so, and this is, if any of you are eating while listening to this, you might want to stop. So back then, what they thought was that these pus-soaked rags that they would have would be healing rags, and they would reuse them on other individuals and other cuts. And so pus was a very prevalent healing tool back then. And of course, they didn't know any better. So, just hold those thoughts aside. So Miescher scientifically wanted to know, okay, so how does pus work? What is pus? How is this healing people? What is this stuff? And so what he did was he started, experimenting and analyzing pus, and he was able to isolate this material. He isolated what he called nuclein from the nuclei of white blood cells, basically from pus. And the thing about this nuclein is that it contained proteins and this molecule, this chemical called deoxyribonucleic acid. And so chemical analysis of it, well, it wasn't known just yet what it was. So chemical analysis of nuclein revealed that it contained carbon, hydrogen, and oxygen. Now, those are things that you would expect, right? Biological organism. But it also contained large amounts of the less common nitrogen and phosphorus, the far less common phosphorus. But the amount of phosphorus in it was pretty significant, far more significant than you would normally find in just general other parts of the cell. And so therefore, the nuclein was of particular interest to certain chemists. Nuclein from various cell types also had the same makeup, so this was kind of interesting. So, well, then what's special about pus if it has this nuclein when other cells have the same nuclein material? And then, okay, so now we'll just fast forward. Many years later, it was shown that the nuclein, so like Miescher didn't, immediately show this, but it was later shown that the nuclein contains the chemical deoxyribonucleic acid. Now, you've heard me say this before, and I know it sounds weird that your genetics professor is telling you DNA. Well, no big deal. It's a chemical molecule. It's been around forever. You've heard me say in the past, you've eaten metric tons of this stuff, and you just break it down and reuse the parts, right? You just recycle it. It's not like it's going to change you. You consume, whenever you consume food, you're consuming massive amounts of DNA of different bacterial cells, your own cells from the skin sloughing off in your mouth when you're chewing, to the DNA of whatever it is that might be the material that you're eating. If it has, reproductive material, if it has DNA in it, right, which it probably does, tons of DNA, and you're just going to break that stuff down because it has carbon, hydrogen, nitrogen, oxygen, phosphorus, right? But after it was shown that this nuclein material contained deoxyribonucleic acid, Scientists didn't know that that was some special molecule that nature had chosen, like we have, and it has some exalted state now in science that it didn't have that yet. And so chemists, worked with nuclein and they worked with deoxyribonucleic acid, and that's about it. Nothing really happened there. In 1882, so we'll just leave DNA to the side for now. In 1882, you've heard me talk about the Walters, right? So in 1882, Walter Fleming identified thread. So this is, shortly after, I believe, Mendel's death. And Mendel's died in obscurity. Nobody's really paying attention to Mendel's work. But Walter Fleming identified these thread-like bodies during cell division that he called chromosomes, bodies of chromatin. And so these thread-like bodies are chromosomes. And then, decades after Mendel's death, the other Walter Sutton. There we go. So Walter Sutton linked chromosomes as the carriers of Mendel's unit of heredity, or genes. So now we have a link between chromosomes and heredity. And the thing about chromosomes is that, as I said, they're made up of DNA and proteins. So chromosomes contain DNA and protein. And so now, just, without any sort of scientific experimentation, we can at least, just circumstantially narrow this down to, okay, so the way we pass on our traits is via either DNA, protein, or both, for that matter. It could be both. And so what's the component? Which is the component, the chemical component of heredity? Is it DNA? Is it protein? Is it both? Is it some combination of both? Is it one but not the other? What is it? That's the question that we're up to in the early 1900s. Fast forward, so now that question hasn't been answered, and it's not going to be answered for about 50 years. Okay, but we're going to build up to that answer. So I expect you to remember Miescher and nuclein. I expect you to remember Fleming and thread-like bodies, discovery of chromosomes. Sutton, linking those chromosomes to Mendel's work. Now we're up to one of three experiments in this lecture that you need to know and understand intricately, because I'm going to ask you many questions about them because they are so instrumental in our understanding of how DNA is the chemical component of heredity and protein isn't. Oops, I didn't mean to give anything away there. You probably already knew that, but that's what we're working up to. So the first experiment, first of three, let me just, I'll say them now so you can write them down if you want, so you know what to look for. The first experiment is, so in a normal lecture on the board, I would start making a list, right? And my list would be, all right, 3 experiments. Fred Griffith's experiment, so Griffith. Then there's going to be one that's called the Avery McLeod-McCarty experiment. And then there's going to be one that's called the Hershey-Chase experiment. And so I would write those on the board. And then we would go over them, and I'd put little check marks by them as they were covered. So the first one we're going to address, and we're going to go through them in chronological order. So 1928, and you can see 25 years have gone by since linking chromosomes to the chemical component of heredity, and we still, Griffith's experiment didn't say, hey, it's DNA, right? So that still hasn't been shown yet. What Griffith did show was, okay, so Fred Griffith performed an elegant experiment that increased our knowledge of the chemical nature of genes. And you can see there, increased our knowledge of the chemical nature of genes. Here's what he was doing. And the analogies to present day abound. But basically, he was working with, so back in the early 1900s, the pneumonia was a major cause of death. One could even, basically, I don't know if it reached pandemic state. I would have to look that up. It was officially recognized as a pandemic, but it was certainly a very prevalent killer. And so Fred Griffith wanted to come up with a vaccine for a form of Streptococcus pneumonia that was fatal, so that you could be vaccinated against it and therefore have a better chance of survival. And so 2 strains of Streptococcus pneumonia, or pneumoniae, some people say, a bacterium responsible for a form of pneumonia, were used. And so they are essentially going to be what's called the rough strain and the smooth strain. So the smooth strain is virulent. It's the dangerous one. It's the killer strain. That's because it's virulent. That's what that means. And the reason why it's so dangerous is because it's the smooth strain. So it had a gelatinous coat on it. Just imagine that it was slick. It was slippery. It was very difficult. So remember, bacteria, viruses, they evolved to pick up mechanisms to avoid our immune system. And so that's the predator-prey relationship between us and pathogens, right? So we have an immune system. Our immune system's constantly trying to fight these things off. There are always novel viruses or different types of bacteria that pick up some sort of advantage where they can evade our immune system, and then they become more virulent, which is good for them because they can infect us, they can replicate in us, they can use our resources to replicate and then be spread, even at the cost of our own life. So the virulent smooth strain had that gelatinous coat on it, which made it very difficult for our antibodies to bind to it. And so that's why it was more difficult for us to clear it from our system, and therefore it would be able to get a better threshold, and then be the lethal strain. So that's the smooth strain. Alternatively, the less virulent rough strain lacked that gelatinous coat. Our antibodies could bind to it more efficiently. And so, yeah, you might get a little sick, but your immune system could fight this one off. So R strain is going to make you sick, but you can fight it off. You can survive. S strain is the fatal strain of Streptococcus pneumoniae. So now Griffith was working on a vaccine. So let's talk about how vaccines were made back then. So nowadays, vaccines are made as subunit vaccines. So you know that some people think that, oh, they get a vaccine, oh, I got sick from the vaccine, but I'll be okay, right? No, it's possible, ever so slightly possible, that at the site of injection, I shouldn't say slightly possible, it's possible at the site of injection that, you have a little bit of raised area, a little redness and that sort of thing. But you're not going to get the disease that you are being vaccinated against from the vaccine. That's because nowadays, let's say we're vaccinating against a particular virus. What you do is you take the genetic material within that virus, you analyze it, you pick out the gene or any sort of coding region for just a small subunit of the protein coat of that virus. So not the virulent DNA or RNA or anything like that, but just imagine the packaging, right? So I'll use the SARS-CoV-2 virus because that's in the news constantly. So the SARS-CoV-2 virus that's responsible for the COVID-19 disease, that's responsible for the global pandemic of 2020, it has viral spike glycoproteins. It's got these little trimeric proteins, these little spikes on the outside of the virus particle, which makes it really, really contagious because these little spikes are really good at sticking to stuff. So if you were to design, and I'm just speculating here, I don't know what the actual researchers are. Well, actually, I've read a few of their work, but of course they're all racing against one another, so they're holding things pretty close to the chest. But if you were to design a subunit vaccine against SARS-CoV-2, one strategy you could use is to figure out what codes for that viral spike glycoprotein, and then just take that gene. Let's say there's a little bit of nucleic acid that codes for that. Take that sequence and then make up a tremendous amount of that viral spike glycoprotein. Make just that protein, nothing else. So it can't infect you. I mean, it's basically just going to be the protein. And then that's your subunit vaccine. So you make a ton of that, and then you inject that into individuals. Then their bodies, basically recognize this foreign substance in their body, and they say, oh, that's foreign. I'm going to make antibodies against that. And your immune system has now a response to that spike glycoprotein. Then when you get exposed to the real virus, which is coded in that viral, spike glycoprotein, your body's like, oh, I know what those are. I have antibodies for that. You can make antibodies against it, clear from your system. That's how we do it nowadays. But as you know, not everybody understands how vaccines work, obviously. And so some people think that you can get sick from the vaccine. And I'm not talking about like anti-vaxxers and their falsifications about autism being linked to vaccines, which it's not. No scientific evidence whatsoever for that. What I'm talking about is like you get a flu shot and then you get a mild version of the flu afterwards, and then you're better, right? You like feel run down for a day or two because you got a flu shot, and so obviously you must have some mild version of the flu. That's not how it works. But that thought process comes from the old way we used to make vaccines, like, for example, the way in which Griffith was trying to make his vaccine. Back in the day, you would, and I would not recommend this, you would grow up in a laboratory a tremendous amount of the very virulent thing, the very thing you were trying to vaccinate against. You'd make a ton of it, which obviously one accident, and that could go very wrong. Then you would simply heat kill it. You just boil it. Hopefully you killed everything in there. In the case of Streptococcus pneumoniae, hopefully you lysed every single cell. And then the lysate, the parts of the cell that you're trying to make a vaccine against, you'd mix that up, and then you'd inject that into people. And, well, that's the theory. That's what was done originally. And, hopefully none of those cells are still available or alive, and so you're just injecting parts, and so then your immune system can make antibodies against those parts, and when you get exposed to a virulent bacterium in this case, then you already have the ability to make antibodies against it. So, that being said, I'm sorry, that was a little long. That being said, Fred Griffith was trying to basically create a vaccine for Streptococcus pneumoniae. So he had, or pneumonia. So he had a number of controls he was going to do. Here's his plan. He's going to make a vaccine. That vaccine is going to have heat-killed S, because remember the S strain is the virulent strain. You don't want to give anybody the live version of that, so he's going to boil that stuff. But if you put that into somebody, it's just a bunch of parts, and you're not going to get a very aggressive immune response. So he wants to make sure that it's a good vaccine and that it's going to really give you a nice aggressive immune response against that stuff. So in addition to the heat-killed S, he wants to add to his vaccine, and this was perfectly normal practice back then, he wants to add to that heat-killed S a few, small dose, of the less virulent R strain. So yeah, you might get a little sick, but your immune system will be able to handle it. You will get better. And the theory is you will have a nice, strong immunity to both the R and the more virulent S strain. And so, if that's your idea of a vaccine, you should do some controls beforehand before you go start injecting people, right? So here are his controls. So first, in this first part of the experiment, what he did was he's like, okay, well, he's good. Remember, he wants to inject live R along with his heat-killed S. So we should make sure the live R doesn't kill off the host. And so we're going to use mice. And so mice injected with live cells of the less virulent R strain, they get injected. The mice do not die. They do not, eventually they clear it from their system and there are no live R cells in their blood. Okay, so that control says it looks like it's okay to inject live R. The less virulent strain, the rough strain, we don't expect that to be a killer strain. We want to include that in our in our traditional vaccine mixture. And so we want to make sure that if we include it, by itself, it doesn't kill anything. We're good. That's what this control says. The next control is, okay, mice injected with live cells of S. The reason why we're doing all this is because the S strain is so virulent that it can be fatal. Let's test that. So you inject some poor mice with some live S strain. The mice do not you can see the little mouse goes to sleep, never wakes up. And so the mice do not clear from their system. They cannot survive it. They die. Mice die. You take a little post-mortem blood sample, and what you find is that there are live S cells in their bloodstream, meaning the Streptococcus pneumoniae is live and well. The S strain is live and well, so it is basically responsible for their death. Whereas over here, the live R strain can be cleared from their system, no live R strain in their blood after infection. Okay. All right, so that means, okay, S strain bad, so we should heat kill the S strain. So mice injected with heat-killed S cells are then tested, and as you can see, the mice cells do not die. There is no live S cell in their blood, and so blood samples is what I was thinking, right? Not like Crips and Bloods. And so essentially, there is no live S cells in their blood, and so that tells us that if we were to inject a mouse with live R, its immune system can clear it from the system, no live R strain in the blood. If we were to inject a mouse with heat-killed S, The mouse can, basically survive. It clears anything from its system. There's no live S cells in their blood. So if we mix these two things together, then we should have a viable vaccine. And that's the goal. And so he did that. And it didn't work. Right? So mice injected with live R cells plus heat-killed S cells died. And here's the crazy thing. Here's the, like the moment at which I joke with my students in class. I'm like, okay, everybody get ready to gasp, right? Because this is the moment of like, oh, the twist. And so, and then I'll just say what's going to happen. So I tell them about what I'm about to tell you. And then usually nobody gasps. And then I give everybody hell for not gasping because you should. And so then facetiously people gasp, and it's a great time. But I digress. All right, so what we have is mice, end up dying. Then you take a little postmortem sample like we did here, and you look at the blood, and here's the crazy thing. Here's where you would gasp right now. You find live S cells in their blood. Right? I mean, we could be silly about this, and we could say that you find live zombie S cells that were dead and have been brought back to life. This is a form of bacterial resurrection, right, and all sorts of stuff. But fortunately for us, Griffith was a scientist and didn't realize, he realized, yeah, okay, that's science fiction. That's not what happened. And so Griffith isolated live S-type bacteria from mice injected with heat-killed S and live R cells, which you shouldn't be expected, because the heat-killed S is destroyed. There are no remaining S cells that are just like dead but still intact. They have been obliterated into little tiny parts, right? So there's no resuscitating one of these bacterial cells. But there are live R cells in there. So he theorized that there must be some sort of transforming principle. There must be some physical, chemical molecule or molecules that are floating around because we blew up all the S cells that somehow get into an R cell and transform it into an S-type strain. So he hypothesized a transforming principle from the S strain, some sort of little, molecule or molecules or something like that, from the S strain was responsible for the conversion of R strain to virulent S. Now, if you think about it, you might see why I'm bringing this up, because that means that there's some sort of chemical component of, in this case, not so much heredity from one generation to the next, but something that encodes different aspects of life is perhaps picked up by the R strain, and then that R strain becomes transformed into the virulent S strain. Now, this is the end of the Griffith experiment. He basically said, hey, look at that. There's a transforming principle. Don't know what's going on there. I'm going to get back to work on my vaccine. Here, scientific community, you figure it out. So he published this, and that was absolutely a major publication in terms of, our quest for understanding heredity. There's a link here that needs to be made. Like, what is it that can change an entire R strain into a different strain of Streptococcus pneumoniae that then makes it more virulent? Because obviously we want to know what that is, because if we know what that is, then we could actually perhaps, in his case, we could think of different ways to prevent that. But more importantly, just generally in basic science, we have like help determining whether we have some sort of insight into hereditary material. Okay. So here's just, I apologize for spending so much time on that last slide, but, let me just quickly summarize it here. What you've got is live S-type bacteria were injected into the mice died. Live R-type bacteria were injected into a mouse survived, doesn't have any bacteria in it. So it clears it from its system. Heat-killed S, injected to a mouse survives, doesn't have any bacteria in it. Heat-killed S plus live R injected into a mouse dies, and then you can see, if you do a post-mortem, you take a little sample and then you put it on a plate, live S-type bacteria were isolated from the dead mouse. And so I like this image. I don't believe it's from your book. I think I got it from a different image, different text. But Or I might have that backwards. This might be from a book. But the point is, I like this image because it's a quick little summary of Griffith showing that there must be some sort of transforming principle. Because you got dead cells, they don't do anything. You got dead cells, they don't do anything. You got a live cell. Maybe some part of the dead cell got into the live cell and turned it into these, live S-type cells. Okay, so although Griffith showed evidence for transforming factor, he didn't pursue that, and it was still unclear whether DNA or protein was involved. Because let's be frank, I mean, you just blew up cells, so that's every part of the cell in there. There's lipids, there's carbohydrates, there's nucleic acids, there's proteins, there's everything in there. So that was 1928, right? Years go by. People start checking, well, is it this? Is it that? They fractionate cells. They basically separate cells into their different components. And they try to see whether this is important, this is important, this is important. And they start to eliminate certain things. So now we fast forward to 1944. And we get to the Avery, McLeod, and McCarty experiment. And basically this one is, so now a lot's happened since 1928, but they basically extended Griffith's investigations by mixing R strain with DNA. So at this point, well, let me give you a little bit of background. At this point, sorry, I think I cut out there. So let me try again. So at this point, what I'm trying to convey is that there are two camps. All right. So right around 1944, there's two camps. There are the camps. So lipids have been, eliminated by experimentation. Nobody thought it was lipids. So basically, there are a group of scientists that think that the chemical component of heredity, we're getting close, and the chemical component of heredity is either proteins or DNA. And many, many people believed it was protein, because proteins are composed of 20 different amino acids. They're so complex. That is a much more suitable mechanism or molecule for encoding the complexity of life that we see throughout the entire natural world, as opposed to DNA, which only has 4 bases. So, I mean, you could only make, like, how big is your alphabet? You have an alphabet of 20 letters. How many words could you make as opposed to an alphabet of 4, right? So how could you encode the diversity of life with just 4 bases, right? This was a time before we really had an understanding of, just think of binary, right? Ones and zeros, how much information we can store with just that. 4 is plenty, but back then we didn't know that. And so there's quite a few people that believe that proteins are the chemical component of heredity. Avery, McLeod, and McCarty were in the group that believed that DNA was the chemical component of heredity. In the minority, actually. And so they extended Griffith's investigations by mixing R strain with the DNA extracted from S-type bacteria. They got the same result. So here's the thing. So this would be, I mean, these are, I'm sorry, they're a little pixelated, but this is just simplified explanations of Griffith experiment. So here you can see live, non-encapsulated, non-virulent bacteria, so the R strain. Here's heat-killed viral or virulent bacteria. So you mix those together, and so a mixture of heat-killed virulent, so heat-killed S and live R bacteria. Put that in a mouse. Mouse flips upside down, because when mice die, they just flip upside down. Right? I'm just kidding. Obviously, they don't always do that. They rarely do that. But basically what happens is, okay, the mouse dies. Here, Avery, McLeod, and McCarty were able to take, okay, so the live R strain, non-virulent strain. Then they took some ramen noodles, right? So they basically said DNA isolated from heat-killed virulent bacteria. So just the DNA, threw that in there. Then they, basically just mixed it up. And then they injected it into the mice. The mice died. And that's strange. And so they found that the bacteria became encapsulated, meaning they went from R to S strain just by adding DNA, which to a logical and objective observer would say, that looks pretty obvious that DNA is the chemical component of heredity, because it just caused you to change your live R strain into live S strain. Just by adding the S strain bacteria's DNA, the R strain can be converted into S strain. And so, nowadays we would say, sure, you introduce the DNA from the S strain, that changed the expression of the R strain such that it mimicked it now had the capacity to have the same gene expression as the S strain, be virulent, and therefore it is now virulent, right? But of course we didn't know all of that back then. And so now this should have been the end of this should have been, okay, DNA is the chemical component of heredity. However, remember that the protein group was a very large group of scientists, and they were convinced that it was protein. And so they're, peer reviewing this, they're looking at this critically, and they find flaws. Now, in my opinion, these are, quite a stretch, but they find flaws with this experiment. So the controls for this experiment that Avery McLeod and McCarty performed, basically, has proteases in it. And so one of the controls was, okay, so we took the DNA and we treated it with protease to kill all proteins, to destroy all proteins. Proteases are enzymes that chop up proteins. Proteases are the things that we have. They're secreted by your pancreas. We use them in digestion to break down stuff. So, many organisms have proteases. Proteases destroy proteins. And so we're trying to say, is it DNA or is it protein? And so in one of their controls, they thought they'd include proteases. Proteases did not prevent transformation. So this is your way of saying, okay, so if we destroy all proteins, DNA remains intact. That didn't prevent transformation. However, if we introduce DNase, which is an enzyme that destroys DNA, DNase and not protease, so proteins are intact, but DNA is not. DNase will abolish the transformation. So when you introduce the DNase in that particular control, the DNase abolishes transformation of R to S colonies, meaning DNA, you conclude, okay, so DNA must have been useful. DNA was Griffith's transforming principle. And reasonably, this is sufficient. And it's unfortunate that Avery, McLeod, and McCarty don't get the recognition that they deserve for actually showing that DNA is the chemical component of heredity. But that's because the protein camp, while grasping for any reason to still believe proteins, made the following argument. And usually I ask the class at this point, so what would you argue if you really thought it was proteins and you're looking at this and you wanted to say, nah, I'm not convinced. How could you scientifically make an argument that there's some flaw here? And we go back and forth in class for a little bit, but then people come to the realization that, hey, their controls involve adding proteins, right? Proteases are proteins. DNase is a protein. So here's why I say it's such a stretch. What are the chances that the proteases or the DNase that you added as a control is sufficient to be a chemical component of heredity? I'm thinking pretty slim, but that's the argument. The argument is your controls are flawed. You're still using proteins. And so how on earth could we ever convince the protein camp that it's DNA that is the chemical component of heredity and not proteins? The best way would be to actually do an experiment in which DNA and protein were the only two things. And you could actually see, okay, I'm going to take away the protein and leave the DNA. That worked. If you get the same outcome as what's on this slide. So I take away the protein, leave the DNA, and that works as a transforming principle. Take away the protein and, I take away the, I've lost track. So let me start over. I take away the protein, leave the DNA, shows that, and the, cells are transformed. I take away the DNA, leave the protein, cells are not transformed. The first experiment that I mentioned in my, third attempt of saying it is take away the protein, leave the DNA. If that shows a transforming principle, that demonstrates DNA is the transforming principle. The second experiment, take away the DNA, leave the protein, nothing happens. That would show that protein is not the transforming principle. That would be the best way to show this. And in fact, that is how Alfred Hershey and Martha Chase were able to demonstrate this. And their experiment deserves a tremendous amount of credit. So let's talk about that. Oh, I'm sorry. One last thing before we get to the Hershey-Chase experiment. I forgot about this little portion of the slide. DNase is, we already talked about how DNase abolished transformation of R to S colonies. Here's a visual, right? So here are type R cells, and, the mice are able to clear that from the system. So you take a little blood sample, you try and grow up some type R less virulent bacteria, you're not going to get any. If you take type R and type S DNA extract, mix those together, you do get transformed type S virulent bacteria. you go through the process, right? You inject it into mice and blah, 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 and then there you go. Over here, if you have type R and type S, DNA extract plus DNA switch destroys the DNA, you can see nothing happens. That argues that DNA is the transforming principle because when you add DNase, no transformation occurs. This is type S DNA extract plus RNase. You do that, okay, so RNA is not involved. You add protease to kill off proteins, and proteins are not the transforming principle. So it sure looks like DNA, not RNA, and not protein are the transforming principle. Okay, now let's look at the Hershey-Chase experiment. I think what's important to start with is the description you may have heard before in science, where you have an elegant experiment. And I don't know if I've said this before in class, so forgive me if I'm repeating myself, but when you use that adjective elegant, that's not just a regular old adjective. You're not just saying, oh yeah, nice job. When you say an elegant experiment in science, in general, in almost any field in science, you are describing an experiment that is exemplary. It is elegant in the sense that its controls are bulletproof, its conclusions are basically as close to indisputable as you can get. It is an extremely well designed, well thought out, and simple. It's very complex, but it's so complex that the results are simple. They're easy to understand. It is an experiment that shows you something scientifically in a simple, straightforward manner. That would be an elegant experiment. And it is, the Hershey-Chase experiment is one of those experiments, not just because we're talking about genetics and biochemistry, whatever. In any field, there are certain experiments that are of a upper echelon of elegant experiments. And I got to say, and I know I sound like I'm geeking out here, but the Hershey-Chase experiment is absolutely brilliant. It's an elegant experiment, and it's actually also the way that we were able to determine that DNA and not protein is the chemical component of heredity. So I've said this before, because of the complexity of proteins and the simplicity of nucleic acids, proteins were the ones that people thought were the chemical component of heredity, and DNA wasn't accepted as the genetic material, the chemical component of heredity, until 1952. And what happened then was basically the Hershey-Chase experiment became widely known. Okay, so Alfred Hershey, Martha Chase. There's some more names you need to remember for their experiment, right? We have the Griffith experiment, we have the Avery McLeod McCartney experiment, and now we have the Alfred Hershey, Martha Chase experiment. So they performed this ingenious experiment, right? And so here's what they did. They took advantage of, they were experts in a number of different fields. So they were very well-rounded scientists, and they took advantage of their knowledge, their expertise in virology. They were dealing with bacteriophage, which are viruses that infect bacteria. You could see that here in the schematic, right? So what we have is a bacteriophage. So these little bacteriophage are very famous, actually, in Hollywood. So look closely and you can see that shape, right? So this is not a very good rendition, but an icosahedral head, a little cylindrical body, and then these little spider legs, right? And there's usually a lot of these spider legs. And so they basically kind of sit down on side. This is not drawn to scale. These things are tiny compared to an E. coli cell, for example. But they sit down on the outside. I'll show you an image in a second. And they inject their genetic material into the cell, cause the cell to lyse. And when it lyses, it explodes. It basically spills out more virus particles, so now you can infect the surrounding cells. And I said they're famous because think of all the different Hollywood like aliens and different in video games and whatever. That shape is characteristic. You see it all the time in all sorts of different movies. And so I don't want to go too far off onto a tangent, but a lot of what you see in movies are actually obscure biological things that are already here on Earth. Like, for example, I mentioned aliens, right? So the actual movie Alien or Aliens, that crazy alien that has the acid for blood and all that sort of stuff, that one. So that one is actually, you can find it here on Earth. It's obviously not, from space and has acid for blood or anything like that, but it's this little tiny organism that lives in the ocean, and it's actually super cute. It's teeny tiny. I mean, it looks scary as hell up close because it looks like the alien. It even has a little thing that comes shooting out and everything like that. But again, I digress. So let's go back to this. So what we have is bacteriophage. All right. So now these two people are pretty smart, or were, and they knew a number of different things. Like I said, they understood how phage work. And so they thought, okay, so already you have a system where you could possibly transform cells, right? You could kind of mimic Griffith's experiment. They also knew that bacteriophage contain only DNA and protein and nothing else. So if you're trying to answer the question, which is the chemical component of heredity, DNA or protein, bacteriophage are very well suited to be, your experimental system for that. On top of that, here's where it gets even more brilliant. It's absolutely amazing. So they were extremely knowledgeable in physics and radiochemistry. And so let me say this. DNA, you might recall a long time ago, I might have actually mentioned this. DNA contains, not too long ago, just a few slides back, I told you DNA contains carbon, hydrogen, nitrogen, right, oxygen, and then the less common phosphorus. I say it's less common because the phosphorus that you find in DNA is not present in protein. Protein does not have phosphorus. Alternatively, vice versa, if you will, proteins contain sulfur. DNA does not. So Hershey and Chase exploited this difference in an absolutely masterful way. What they did was they decided to radio label. So this is the 1940s, 1950s, right? It's not like you had, like super powerful electron microscopy that we have now, easily readily available in computing power and all that sort of stuff, rendering your digital image and all that. So they needed a way to follow the equivalent of following these molecules, the phage particle and its nucleic acid, following it with their naked eyes, right? And obviously you can't follow it with your naked eyes. You can't see it, it's too small. What's the next best thing? Radioactivity, because you can trace radioactivity quantitatively. You can follow it quantitatively, and for a very long time, radioactivity has been utilized throughout diagnostics and all sorts of different aspects of biochemistry and whatnot to radio label things and follow them. And I've done it many times, so it's not an old obsolete technique that was only done back in the 50s. Nowadays, absolutely, you want to avoid it. It requires a tremendous amount of expense in terms of training and disposal and hazardous waste pickup and maintaining your radioactive waste, even being licensed to have it in the first place. And so you want to switch to, like, nowadays we switch to a lot of fluorescent markers instead of radio labeling things. But back then, you didn't have all those fluorescent markers yet, so it was radio label. And they radio labeled the DNA, which contains phosphorus and not sulfur, with P32, a radioactive isotope of phosphorus. They radio labeled the protein with S35, a radioisotope of sulfur. And like I said, this was masterful because on top of the just knowing, hey, well, there's phosphorus in DNA and not in protein, and there's sulfur in protein and not in DNA. On top of that, they use two different emitters. If you know anything about radiochemistry, you may have learned about alpha emitters, beta emitters, gamma emitters, electron capture, basically, what is the other one? Positron emission. So there are different forms of radioactive decay. They're measured in different ways, and they are quantitative using different instruments. So P32 is a beta emitter. It emits these little tiny, basically electrons, high energy electrons, whereas S35 is an alpha emitter. It emits helium nuclei, two very different forms of radioactivity. So not only could you have both of them present in the same cell or same experimental system and still be able to tell where the DNA is and where the protein is based off of the radioactivity. It would be a little harder to do it when they're both together. You're going to have, it's going to be a little more difficult. But the point is, by the type of radioactivity, you can tell whether you're looking at radio-labeled DNA or radio-labeled proteins. So I mentioned, so you need to know understand the way in which phage sometimes work, right? So they, like I said, sit down on the outside of the cell. They inject their genetic material into the cell, and then the phage particle is the protein portion of the phage particle is lost. So the protein never goes into the cell. So if you want to know if DNA and not protein is the transforming principle that Griffith identified all those years ago, right? We're talking about 20 something years ago. What was that? What was it? 1928 to 1952, I think it was. So let's say 24 years ago. Did I do that right? Yeah, I did. Okay. So basically, this is perfect. Right? This is a perfect experimental system for doing it, and they chose that. They came up with it. They thought of it. So obviously, elegant experiment for them. And so let's take a look. So here's just an image of a poor little cell that has been ravaged by phage particles, right? I mean, it's not even a cell anymore. It's already lysed. This is just a portion of the membrane of the cell. And you can see all the phage particles that are on it, and it's already lysed and expelled all the extra. You can see there's like little parts of phage particles in there, but it's exploded and lost all its phage particles. Okay, so as I said, here it is in bullet points. They used S35 labeled viral protein and P32 labeled DNA to follow viral infection in E. coli. And so here's what they saw. And what I need you to do is be able to make the proper conclusion from these results. So I will tell you what happened, then you want to try and come up with your proper conclusion, your proper scientifically accurate conclusion. Do not add on any facts. Just do the minimum, basically state the minimum that you can conclude from this experiment. So here's what, they did multiple versions, right? Multiple, like sometimes the protein was labeled, sometimes the DNA was labeled, etc. So let's look at one scenario. So one of their controls, one of their portions of their experiment is that the virus particle labeled with S35 and the DNA not labeled with P32 is allowed to, so the way this works is you grow the virus particles up in medium containing either S35 or P32. If you do that, then you're going to create radio-labeled virus particles, and if it's only grown up in S35 containing medium, then the protein coat is going to contain the radioactivity. The DNA will not. If you grow it up in P32 medium, opposite. The DNA will be radioactive. The protein coat, the viral particle, is not going to be. In this case, they raised viral particles in S35 containing medium. Then they took a small sample of that, and then they allowed that to, they aliquoted that small sample into a growing culture of bacterial cells, so E. coli cells. They only allowed the virus particles to be present in that culture of E. coli cells for a short period of time, just long enough for the virus particles to infect some of those cells. Then they centrifuged it, right? So you can see you add the two together. This is what's happening on a microscopic level that we can't see. And then the S35 containing viral coat protein particles, those are on the outside of cells, right? So we want to separate those away. We want to remove those. So what they did was centrifuge the culture. If you gently centrifuge the culture, the viral particles are tiny compared to the cells. So the cells are going to pellet out. They're going to basically, if you centrifuge, you spin the culture down. Cells are going to pellet out. They're going to accumulate at the bottom of your centrifuge tube, form a pellet, as we call it. The viral particles, on the other hand, are going to remain in the fluid, which is called a supernatant. And so, we call it soup for short in the lab. And so you could basically separate out all the, like if you see in any news coverage of the laboratories working on a vaccine for the SARS-CoV-2 virus, they tend to show the most mundane things because it looks awesome, right? And so they show, I was watching something today and I saw like like little conical centrifuge tubes, which are basically like just plastic tubes with a little bit of liquid in them. But they were showing those as if they were like some instrument of higher science, and they were just sitting there and they were being spun gently. And so it was it was just basically they were just agitating them. So very mundane thing, but it was shiny and looked cool. But basically what happens is you spin down these cultures, and then the viral particle, the protein part, remains in the supernatant. So you pour off the soup, and what you're left with is a pellet. So you resuspend that pellet in regular medium, and then you just let it grow and see what happens. And in all cases, these E. coli cells then turned into virus producing factories, and they lysed open and viral particles were obtained. So this experiment, what can we conclude? we can conclude that this experiment demonstrates protein is not the chemical component of heredity, because we can follow the protein. We know exactly where the protein is. The protein is radio labeled. We know that if you allow the protein to have access to the cells, but then take it away, the cells are still transformed. And all of the protein is gone. You can't argue that, oh, well, maybe a little some of it got by you. Nope, it's been radio labeled, and you can quantitatively measure that. And so that was the thing that Hershey Chase added to this. There's like, no, 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 you can't. No, this is bulletproof. We can quantitatively measure how much we started with and how much we ended with. We got all of it, and yet all of those cells are turning into virus-producing factories, and they're lysing open and spilling out phage. So something transformed them, and it's not the protein, because I have all of the protein back right here. I collected it all by centrifugation. So this aspect of the experiment shows protein is not the chemical component of heredity. Then if you switch your radio label, and so now we're looking at P32 labeled DNA and not protein. The protein's not radio labeled. P32 labeled DNA was basically found inside the cells. So here's what happens. All right, we do the exact same experiment. We grow up the phage in P32 label, or just P32. Right, you give them the access to the radioisotope P32 in their medium. They incorporate that into the DNA that they're making, because they're not going to incorporate that into proteins, because it's not in proteins. And so then you take these radiolabeled phage particles. Again, you allow them, you take a little aliquot, squirt it into your culture of E. coli cells, rapidly growing, logarithmically growing E. coli cells. Give them just a minute or two, and then spin that down and pull off just exact same protocol as you did with the previous example. So everything's the same. So you've already demonstrated that this protocol will remove all of the viral protein. And so you remove all of the viral protein and you watch what happens. The E. coli cells are radiolabeled, indicating the viral particle transferred its DNA, its radiolabeled DNA, to the E. coli cells. Then the E. coli cells go on to lyse, produce phage, spill out particles that are actually radio labeled too. And that tells you, okay, so now this aspect of the experiment, the Hershey Chase experiment, shows that DNA is the chemical component of heredity and not that's it. I should say that. It just shows this demonstrates DNA is the chemical component of heredity. The previous one demonstrates protein is not the chemical component of heredity. Combined, these experiments show us that DNA and not protein is the chemical component of heredity. And that was a big deal, because there were quite a few people still in the protein camp. Not everybody, but there were quite a few people still in the protein camp because of just the logic, right? I mean, proteins are so much more complex than, they have 20 different amino acids. They have just what seems like an infinite number of different configurations, whereas DNA is going to be a long chain molecule that is basically composed of either A, C, G, or T in some sort of fashion. Like, you just have 4 characters of code as opposed to 20 characters of code with the 20 naturally occurring amino acids. So now Hershey and Chase have put the nail in the coffin for the protein group. And what's nice is that they weren't alone in their thought that DNA was the chemical component of heredity. So there are other scientists that were working on, trying to decipher DNA and everything at that time. And they were kind of in the background, but now they're moved to the forefront. So here's just a quick visual, right? Remember what I was saying? So you have your phage particles. You grow them up in either medium containing S35 or medium containing P32. This labels the protein, the viral protein. This labels the, the DNA. Then you give them access to E. coli cells. So I guess this is actually giving them access to the E. coli cells, because we're already mixing in the E. coli cells. So they're already pre-made. And so then they infect. Then you remove the viral proteins, right? There's where, blending and centrifugation. Wow, I didn't realize how hokey my clip art was. And so then what you get is basically, so this is supposed to represent agitation. You got to make sure that the virus particles are off of the cells. So you agitate them using, I would think, a vortex genie, but we could use a blender apparently. And then you can use, this is a little tabletop centrifuge because you don't have to spin it very hard. And so now you've got, the viral proteins removed. You can see viral proteins removed. On the left side, we've got the viral proteins labeled. So all of the radioactivity is recovered. So that means that there's no protein, and yet phage are reproduced. So protein is not the transforming principle. Over here, the viral particles are removed, and the radiolabeled DNA remains, and phage particles are produced. So that argues, this side argues that DNA is the chemical component of heredity. So DNA, combined, the two, DNA is, and not protein, is the genetic material in bacteriophages. It is the chemical component of heredity. That's what this says. These experiments prove that nucleic acid is the genetic material and the chemical component of heredity. So what I have here is a video demonstrating the Hershey-Chase experiment. And so I hope that you'll be able to see the video sometimes, depending on your machine. When I click play, during the rendering process of the video I'm making, the video within a video, it goes blank. So what I'll do is I'll make sure and post this video on our course website as well, so that when I hit play here in a second, if you don't hear or see anything, obviously just continue with the lecture and you can go to the course website, you can watch the video. But let's give it a shot.
In the Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein cover. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacterium. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed.
That's
It
my
did not interfere with production of new phage in the cell.
shake. It's my favorite
In
part.
the second part of the experiment, phage were produced in a medium containing p32 labeled deoxyribonucleotides. This resulted in a phage population with p32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacterium, the p32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacterium. This demonstrated that the DNA, not the protein, carries the genetic information for a new generation of phage.
All right, so now we know the DNA is the chemical component of heredity. That brings us to 1953 and some names you've probably heard. Maybe some names you haven't, but definitely some names you've heard. And so you've probably heard of Watson and Crick, right? The two scientists that elucidated the structure of DNA. Well, let's talk a little bit about how they were able to elucidate the structure of DNA. And I'd like to take a moment to give Rosalind Franklin her props. Because if you think it's bad now, imagine if you were a woman in the 50s in science. So Rosalind Franklin, now she's getting more recognition, obviously a half century later. Oh, more than that, actually, right? But at the time, she was not really well recognized for her contributions. So let's talk about her contributions before we get to Watson and Crick. So Rosalind Franklin was an absolute genius when it came to a technique known as X-ray crystallography. X-ray crystallography is, I'll simplify X-ray crystallography if you're not familiar with it, very much so. So here we go. Imagine I had, usually I do this in class. So imagine I have my hand in front of the board, and I put a flashlight and I shine a light on my hand, and you can see the shadow on the board. So using visible light, which is a form of electromagnetic radiation, that light is going to then travel, hit my hand. I'm going to block out some of the light with the palm of my hand. And then as the light travels between the apertures between my fingers, there's going to be diffraction. And so, because you can look at the double slit experiment or whatever, but the light is going to change direction slightly. And so if you look at the shadow on the wall, you are not going to see, you'll see a negative image of my hand, but it's not going to be a perfect negative image because there's going to be actually a couple little extra fingers if you look closely enough. Each finger is going to have at least, two fingers, and they're just offset a little bit. And there's going to be alternating light spots and dark spots, right? That's because of constructive interference, destructive interference. Now, if I wanted to look at something way smaller than my hand, like a molecule of DNA, I have a crystallized version of DNA. Like I've made so much DNA, purified it, and crystallized it that I have a little crystal made out of pure DNA. If I wanted to shine electromagnetic radiation through that crystal so that the radiation is going to be diffracted and I can have alternating spots, light spots and dark spots because of constructive and destructive interference, that's going to require me to use a much shorter wavelength of electromagnetic radiation because visible light is the wavelength is way too long. You're not going to see any difference. So Rosalind Franklin and Maurice Wilkins, and I certainly want to give Maurice Wilkins his credit too. Rosalind Franklin and Maurice Wilkins, their X-ray crystallography data was absolutely astounding, and it was voluminous. There was so much of it, right? So what you have is, back then you had an X-ray source. You wanted to kind of focus a beam, so you'd have a lead screen with a tiny little aperture. And so then you have your X-rays that have now been focused, this beam of X-rays. You hit the crystal of DNA, and then you get your, you have your old-fashioned X-ray film. And then you would get, areas of constructive interference would hit and make the dark spots. Areas of destructive interference would not expose the film. And you'd have an X-ray diffraction pattern. And from this data, you're supposed to figure out where every atom is in the molecule, which is a daunting task, to me at least. And for all of Rosalind Franklin's work, she did a tremendous, the lion's share of work when it came to X-ray crystallography data. She and Maurice Wilkins had a falling out later in their careers, which is why, she wasn't really recognized for her work, and then later on it gets even worse. Now, she did die before Watson and Crick were awarded the Nobel Prize, I believe, in I'm not sure if it was 1968 or something like that. So she was already deceased, and there, the Nobel Committee says that they don't award the prize posthumously. But these individuals were nominated before her death, and she was not nominated. So it's a very sad story. She should have been recognized far more than she ever was historically, and now she's getting some of that recognition. I just wanted to say she is an absolute savant. Some of her the reason why I'm saying this is if you look at some of her X-ray crystallography data now, it is still superior. And she did this in the 1940s, right, and 50s. With manual techniques, it is still superior. Some of her crystallography data is still even superior to that we can generate now with all the sophisticated equipment that we have now. All right, so we've got Rosalind Franklin and Maurice Wilkins. Then we also, so they contributed X-ray crystallography data. Then we have Erwin Chargaff, another name you need to know. Chargaff, associate Chargaff with base pairing. So Erwin Chargaff he was basically just a chemist. He was just working with DNA. He wanted to know more about the DNA molecule, not because he had any idea that it was going to be so important to nature, et cetera, et cetera. He was just curious about what's this DNA molecule. And he found that anytime you do a chemical analysis of DNA, what was odd was that the percentage of A bases would be equal to the percentage of T. The percentage of G bases would be equal to the percentage of C. And so there must be some relationship between the two of those, right? So A's and T's, G's and C's would be prevalent in the same proportions whenever any sample of DNA was chemically analyzed. Now let's move on to Watson and Crick. Okay, so by no means am I trying to belittle or discount their contribution to humankind. I say this not in that idea. Like, hopefully, you don't even get that impression. What I'm saying is they were not as much the experimental scientists. They were more like an absolutely amazing, brilliant think tank. What they did, which is perfectly legal and ethical, and I am not by any means saying that it isn't. And I'm saying all this. You're probably not aware of why I'm saying all this because some people think it. They say otherwise, right? So Watson and Crick were able to use Rosalind Franklin Maurice Wilkins data, which is published data. There's nothing wrong with that. They read the scientific literature, all right? So they see her data, and they see Erwin Chargaff's data, and then they do a tremendous amount of work themselves to figure out the structure of DNA. And it's very interesting. They had a very interesting relationship. They're more of the I don't know, for lack of a better reference, the Shaq and Kobe of the world, of the molecular biological world. So you can see Francis Crick was more of the you can just see from this image, right? He was playing up to the camera. He was more of the rock star type. James Watson was more of the well, he's still alive. Francis Crick has passed away, but James Watson, is more of the put your head down, go back to work type. Um, and so they were very different personalities, and they didn't have, they didn't seem very fond of one another, and they literally, like they said so. Um, but they recognized and respected one another's abilities, and they needed each other to figure this out. All right. And so I've told every single genetics class I've ever taught that I had the pleasure of meeting James Watson back in 2005, shook his hand, and I won't tell the whole story because this lecture's already pretty long. So, but it's I invited him to join our group for a drink, and it really, in retrospect, and I say this in jest, but it really sound, in retrospect, the way I remember it, he might have thought that I was asking him out on a date, which is certainly not what I was doing by any means. But it's a funny story. So I'm happy in office hours if anybody wants to, hear that, feel free to bug me about it because I come out looking pretty silly, but it was a big deal to me to meet him. This was before, of course, and you can look it up. He voiced very, very unfortunate and prejudiced views later on, and had to resign his. So this was at Cold Spring Harbor. I was there in 2005, and then he later. A few years later, had to resign in disgrace because of his rather unfortunate and not at all scientifically based comments about race and genetics, and it's unfortunate. And then even just recently, he was asked if he regretted those comments, and he doubled down and just reiterated them all over again. I think back in just in 2019, I think it was. So before all of that, right? So had I known what I know now, I probably would have not bothered to, go. I mean, I recognize his contribution to science, and so I'm getting off topic, so I apologize. I'll stop. All right, so now DNA is made up of two. So that we can actually finally talk about the DNA molecule, right? So they figured it out, and so basically DNA turns out to be, as they figured out, made up of two anti-parallel polynucleotide chains coiled to form a double helix. And so we'll talk about what all that means. The scientific community, I've learned, contemporaries of these two, did not have the highest regard for them personally because of, it appears to be a misunderstanding. And that is, I think, so what I've read is that they were trying to be cheeky, as they put it, in their seminal 1953 Nature paper, where you can see it ends with, "It has not escaped our attention that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." That's a hell of a cliffhanger to leave your paper, off on. They thought it was a good way to leave their paper off for set themselves up for the next one, and they thought it was a little cheeky because it immediately suggests it. So if you don't get it, well then figure it out, right? That sort of thing. And it just wasn't received well. And so some old timers that I've worked with, have voiced their displeasure, especially at Cold Spring Harbor when James Watson was present. They were less than complimentary of him, unfortunately. All right, so let's talk about DNA structure. We are actually finally up to the actual molecule. So biological organisms contain two types of nucleic acids, right? They contain deoxyribonucleic acid and ribonucleic acid, both of which are made up of nucleotides. And so nucleotides, let's talk about nucleotides. And so many of you have seen this before, perhaps in, just like 161 or some other, introductory biology course that you may have taken in even high school maybe. And so nucleotides possess a nitrogenous base. These are the nitrogenous bases that are found in nucleotides that are found in DNA and RNA. And so it's a nitrogenous base because it contains nitrogens. And then you also have a sugar. The sugar is a pentose, meaning it is, and if you take an organic chemistry and learn about carbohydrates, you might know it as a furanose. And so this furanose, or this sugar, 5-carbon sugar, has a 5-membered ring in it. And so 1, 2, 3, 4, 5, those are the 5 carbons. And so then you also have the acidic phosphate group, right? So sugar, base, phosphate. Those are the three main components. Three components of nucleotides. Nucleotides are the building blocks of nucleic acids, DNA and RNA. Now, in the past, I'll just tell you what I used to do, and I've learned not to do it anymore. My student reviews have taught me that. I used to joke that, all right, so I need you to memorize all the chemical structures and blah, blah, blah. And I used to tell my students that you need to memorize all the chemical structures. You do not. And then a couple slides later, I would basically say, I was just kidding. You don't have to memorize that. And it was hilarious to me. But I've learned in my student reviews that it's not that funny, apparently, to the students. They did not like that. And so I will flat out say right now, full disclosure, you are not going to be required to memorize every single base structure chemically. But I will ask that you could certainly identify the nitrogenous base from the sugar, from the phosphate, and then a couple of other aspects that we'll point out here in a moment. But the bases are adenine, guanine, and then you have over here grouped together thymine or uracil. DNA has T, RNA has U, and then cytosine, right? So there it is. A, C, G, and T are what you find in, I like to go in alphabetical order. So A, C, G, and T are what you find in DNA, and then A, C, G, and U are what you find in RNA molecules. The sugar is also going to be different. You have, let's start with ribose. This is the sugar molecule you find in RNA, and then at the 2' position. So let's talk about primes. So you're not responsible for having taken organic chemistry at this point. Perhaps you did, but I will say this. If you know anything about organic chemistry, you know that you have to number things to give every atom an address so that you could say where everything's located, right? And so in a nucleotide, you're going to have a ring structure for your base connected to your sugar, which is also a ring structure. So we use the 1, 2, 3, 4, that sort of thing to designate locations on the ring structure in the base. Now we have another ring structure, and we can't use 1, 2, 3, 4, 5 to indicate the carbons because those numbers have already been used. So we use 1', 2', 3', 4', 5' in order to designate the locations of the carbons in that ring. That's just standard IUPAC nomenclature from organic chemistry, no big deal. Now, at the 2' position, so the difference between ribose and deoxyribose is this is carbon 1, this is carbon 2. So at the 2' position, what we see is that we're lacking in oxygen, hence the name deoxyribose. It's ribose without an oxygen. And that difference is pretty profound in the stability of the molecule because there's not going to be a free OH in DNA. This OH is going to be involved in a bond connecting it to the next nucleotide. This OH is going to be involved in a bond connecting it to the next nucleotide. And then this OH is going to be involved in a bond connecting the base. So when this gets, just sitting there as an H, there's no OH there. That makes it a much more stable molecule that's not susceptible to attack because you don't have a polar oxygen-carbon or oxygen-hydrogen bond. So you have partial positive on the H, partial positive on the O. You don't have any of that in DNA, and that is one of the big differences that makes DNA much more stable than RNA. Okay, so you may have heard of purines and pyrimidines. A's and G's are purines, and then C's and T's, or U's for that matter, are the pyrimidines. You can see that here. So we just talked about this. This is what makes this the sugar that you find in DNA, the fact that it's lacking a hydroxyl group at the 2' position. The fact that you do have a 2' a hydroxyl group at the 2' position is what makes this ribose the sugar that is used in RNA. So here is a fully constructed nucleotide, and like I said, I would like for you to be able to identify this is not asking much, I would think. Nucleotide, so there's the sugar, there's the nitrogen to space, there's the sugar, there's the phosphate. So these are the purines. A's and G's are purines because of the purine ring structure that they have. And then here are your pyrimidines, in DNA at least, C's and T's. That's what you find. Now, all of these nucleotides are going to be polymerized, and therefore we are going to end up with those two anti-parallel polynucleotide strands that get wrapped around each other in a double helix. That makes up the fully formed DNA molecule. And so nucleotides are polymerized 5' to 3'. So that's the direction in which they are constructed. And so we can use the primes to give us direction, right? So this is the 1', 2', 3', 4', 5' carbon. So you could see that we go from 5' to this 3' to get to the next nucleotide. So you can see why it's called 5' to 3' because 5' carbon, 3' carbon connects to the next one. So they are polymerized 5' to 3' into polynucleotides via phosphodiester linkage. This is a phosphodiester linkage. So what we have here is ATC, ACCG, just some random sequence that is representative of this polymerized polynucleotide. So there we would write that sequence on paper like this. We would say, 5' end is the starting side, and then we'd go ATC, ACCG, 3' end. All right, so let's talk about this phosphodiester backbone I mentioned. So here you can actually finally see it. And so what we have is, okay, so here's the sugar. And so we've got what is it? So 1', 2', 3', 4', 5', right? There's the labeling. And so if we're going 5' to 3', we're going this way. So we call it a phosphodiester bond because if you're familiar with an ester, C double bond O-R, well, this is a phosphodiester because we have our phosphate group, and so it's P-O-R-O-R, so it's a diester. And, if you're not following, if your chemistry is a little fuzzy on that one, don't worry about it. The point is that the way in which they're linked together is through what's called phosphodiester bonds. And so this outside portion, I say outside, you'll see in a moment why it's called outside, but the outside portion of the molecule is going to be a repeating sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. And so we call that either the phosphodiester backbone or a sugar phosphate backbone. And so the phosphodiester backbone is on the outside of the double helix. The bases are like basically facing inward, and therefore they can hydrogen bond with one another. So now before I advance to the next image, just think of it as a ladder. Right? You've got two rungs of the ladder that are vertical, and so those would be the phosphodiester backbones. And then, I'm sorry, not rungs, two rails, the vertical rails. So two rails of a ladder that are vertical up and down. And so those would be the phosphodiester backbones. Then each individual rung is going to be constructed of a base attached to each rail. And so you have this ladder, and if everything I'm saying doesn't make any sense to you, then this is all stuff I usually draw on the board. Then just think of a ladder and then twist it, right? Like if it was flexible or something, right? Then just twist it. Just twist the image of it in your mind, so you can have a twisted image in your mind. And basically twist the ladder, and then if you twist it a certain way, you can get a double helix. So here's just the phosphodiester backbone schematically. Here are your bases, and then now you can see, okay, here's one phosphodiester backbone, one phosphodiester backbone. Here are the bases. So these would be the rails of the ladder. These would be the rungs of the ladder. A's and T's are capable by their chemical structure of 2 hydrogen bonds between them. G's and C's, 3 hydrogen bonds between them. You might recall I mentioned that when we were talking about gem sustaining and how our genes are more rich in GC content, and so therefore whenever you have a lot of genes on a chromosome, then you don't see as much vivid gem sustaining because it can't penetrate into that chromosome as well. Whereas when there's a lot of AT, you get a lot of dark blue staining because AT only has 2 hydrogen bonds. Well, here you have that ladder, and then, like I said, twist it. And if you twist it in such a way, then you end up with a double helix. So here's just another way of looking at it. So remember I said they're anti-parallel, and so when I say anti-parallel, that means that they're both anti-parallel and complementary. And I don't mean complementary, like so complementary like an I, like hey, you look nice today. Complementary with an E, meaning that one basically nests with the other. And by nesting, I mean hydrogen bonding, base pairing. So here's the top strand. The top strand, we usually write the top strand left to right by convention, 5' to 3', because we read left to right. And the transcriptional machinery is going to transcribe the DNA into RNA when we need to express that gene in a manner that is consistent going 5' to 3' so that it can replicate the top strand. So the top strand encodes the important information, if you will. All right, so when we write DNA, we write it, left to right, 5' to 3' on the top strand. That means that the bottom strand needs to be 5' to 3' in the opposite direction, because remember, these are 2 anti-parallel strands. Anti-parallel. Parallel, it means that they are, 2 lines that are never going to meet, right? And anti-parallel means 2 lines that are never going to meet, but if there were arrows, they'd be pointing in opposite directions. And you can see that directionality here, 5' to 3', so we could draw like an arrow. Here, let's do it. So we can draw an arrow, 5' to 3' going this way, top strand's going that way, and then bottom strand, 5' to 3'. So it's going not very straight that way, right? And so get my laser pointer back. And so basically that indicates that we have anti-parallel polynucleotide strands. Like I said, they base pair. So notice sometimes you get a palindrome, right? Like a race car, right? Same forwards and backwards. So like if it was A, then of course, well, actually, that's not a palindrome. Let me try that again. If it was AAATTT, then, of course, in the opposite direction, it would be 5' to 3', AAATTT. So that would be a palindrome. But most often, then you don't get palindromic sequences. So you can see ACGTG is going to give us a CACGT as the complementary sequence to that sequence, 5' to 3', and this is the 5' to 3' in the anti-parallel direction. So here, let's clean this up a little bit. So basically, there we go. All right, so an easy way to remember it is A's paired with T's. I think I may have said this before. Straight lines go together, right? You see no curves in A or T. So straight lines go together, A's and T's. And then the curvy ones go together, G's and C's. And so we end up with base pairs that are going to be 2 for A's and T's and 3s for G's and C's. I've said all of this out loud, but it's always good to have it in bullet points. So base pairing makes 2 strands complementary in base composition, and they are anti-parallel. Can't stress that enough. So here are some sequence. I probably shouldn't tell you this, but this is how big of a science nerd I am and how much I really enjoy genetics. This sequence is basically what I wanted my license plate to be. But since we're in the state of California, and I'm only limited to, they don't understand genetic sequence, and the DMV. And so I'm limited to 7 characters. I was able to condense this into I needed the CCA, I needed CAT, and I needed GAC. So I overlapped the CAs here. So one of my license plates of my vehicle, so please don't key me, is CCATGAC. And so I'm in stop signs and stoplights all the time. I see in my rear view people like squinting, looking at my license plate, and I can see their mouth like CCAT GAC. CCAT GAC? Like they're trying to figure it out, and it's very amusing. But the reason why I chose this sequence, if you're wondering, it's not just random DNA sequence. There was a lot of thought, way too much thought that went into it, and so I picked it because I wanted a personalized license plate that was something that was personal to me, of course, and so I needed it to be CCA, CAT, GAC. I got CCAC, that's as close as I could get, right? Because obviously there's not enough characters. And the reason why I picked that is because if you go back to the genetic code, actually, we'll get to that in the future. I'm sorry, but if you go look at the genetic code, you'll see that these three triplets encode codons in the RNA, right? And so CCA, CAT, GAC is the same thing as CCA, CAU, GAC. And if you go and look up those codons, you will find amino acids proline, histidine, and aspartic acid. And so the reason why I like those amino acids is because their one letter abbreviation is PhD. And I think that is freaking awesome. And it's my license plate. You can't get it. Haha. I know all of you are going to go fighting to try and get that license plate, but no, sorry, it's already taken. All right, so now that I've confessed to that, so here we have some anti-parallels. Well, here we have some sequence. What's the anti-parallel sequence, right? And so in class, we would normally do a sing-along at this point, and we would go 5' to 3' this way. And so we would say, what base goes here? G. And then what base goes here? T. And then what base goes here? C. And then A, T, G, T, G, G. Right? So G, T, C, A, T, G, T, G, G. There it is. GTC, ATG, TGG, 5' to 3' for the antiparallel strand. So once you have one, you could figure out the sequence of the other. That has profound implications, not just because, Watson and Crick noticed that, but also because it allows us to use one to make another copy of the other. It allows us to repair our DNA when maybe some of it is lost or broken in one strand. It gets a little imagine like some of these bases got cut out for some reason. Well, then, we've talked about deletions in the past, right? So imagine not on a chromosomal level, but on just a nucleotide substitution mutation level. Or actually just a deletion mutation, we have a few of these cut out. We know which ones go there, so we can put them back. So genetic information is stored in the linear base sequence of DNA, and this has tremendous coding capacity, unimaginable coding capacity. You don't have to, you're not limited to just 4 characters of code because you can make the DNA molecules very long. And so then you have 1 of 4 at each position. So, if you think of it, you have 1 of 4 at the first position, 1 of 4 at the second position. You already have in a dinucleotide 4 squared, 16 different choices, 16 different possible combinations. And then if you go up to, 3 nucleotides, well, now it's 4 times 4 times 4. So now you have 4 cubed, 64 different combinations. And remember, our genes, we have things that are thousands and thousands of base pairs long. So there are tremendous different combinations. The sheer number of combinations is tremendously large. So there's tremendous coding capacity in DNA. A DNA molecule, n bases long, base pairs, we measure it in terms of units of base pairs, right? So this one is 1, 2, 3, 4, 5, 6, 7, 8, 9 base pairs long. A DNA molecule, n base pairs long, has 4 to the n combinations. I used to have an exam question where, I said, how many combinations are possible if you had a nucleotide or if you had a DNA sequence that was this many base pairs long? And I didn't think it through. And so a lot of students would just come back with calculator error, right? Because it was just too big of a number. Even scientific notation wasn't sufficient to actually tell them how big the number was. So there is tremendous, tremendous coding capacity in DNA. So the human genome has 3 billion base pairs of sequence. So you go ahead and figure out what 4 to the 3 billion is, and that will be how many different iterations there can be with the human genome. So we could encode a tremendous amount of information. Watson and Crick their model described bDNA. There are different forms of DNA. So bDNA exists when water is plentiful. It's the DNA that's most common. It's the DNA that we're learning about. It's the DNA we're going to assume we're talking about for the rest of the course. However, you must know that different DNA is a chemical molecule, so it's going to take on different shapes, just like a protein takes on different shapes based off of what pH it's in or what salt concentration it's in. DNA is affected by hydration, by salt, by different things. So bDNA exists when water is plentiful and it's fully hydrated. That's healthy DNA. It's the most stable configuration because it's of the basis because it contains water around it. bDNA is the alpha helical DNA that we keep talking about. And so DNA can exist in other configurations. So here's bDNA, nice happy DNA. Then you could also have a reverse helix, right? So you can see the zDNA form. And then over here we just have basically dehydrated DNA. So aDNA exists when there's little water. That's why I call it dehydrated DNA, right? So I mean, if you take a strip of steak and then you make jerky out of it, it's going to get smaller. And so this is DNA jerky, right? So because we're getting very scientific here. And so of course zDNA is going to be, well, remember this is all held together by hydrogen bonding, and you need water to hydrate this molecule. And so if you replace those waters with salts and ions, then you could actually reverse the turns on. So the helix is now a reverse helix, and that's zDNA. So zDNA exists in high salt. And so there's a couple last things for this topic. The ultimate storage problem is the fact that E. coli has one lone molecule of DNA as its chromosome. It's got 4.6 million base pairs of sequence. It's 1,000 times as long as the cell. Here you can see schematic, right? So here's a little E. coli cell, and not drawn to scale in terms of thickness, but length, yes. This is the bacterial chromosome. You need to take all this DNA and wind it up, wrap it up, and stick it into that little E. coli cell. And it can't take up the entire space in the E. coli cell. There's got to be room for other stuff too. You can see here's that poor little E. coli cell that jumped off of a building. And so all its DNA, it's been lysed, and so all its DNA's, this is all one DNA molecule, believe it or not, that it's basically this. That just happens to be spilled out. So human beings and eukaryotes solve this problem by supercoiling. And so each cell nucleus houses approximately 6.5 feet of DNA. If you took all our chromosomes, those 46 chromosomes, stretched them out, laid them end to end, you'd get over 6 feet of length of DNA. Obviously, it's submicroscopic in terms of, the thickness. You wouldn't be able to see them, but they would literally add up to more than 6 feet of DNA, which is a very, very long length considering how small these molecules are that we're talking about. So there's a lot of DNA. And to fit all that DNA into our nuclei, we would need supercoiling. So supercoiling is done by topoisomerase enzymes, and it's just like this. Take a piece of string, take a cord, whatever it may be, wind it up, and let go. And it's going to unravel. So you need to wind it up, 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 you need to keep winding it up so that it gets really tight and compact. Remember, we talked about this with histone proteins way back when we were talking about, I feel like when we were talking about the cell cycle. But basically, if you wrap DNA around histone proteins, that can act as a binding agent, hold that all together. And that's what happens. So each chromosome contains DNA wound around positively charged histone proteins, forming chromatin. And they're positively charged because DNA has all those phosphate groups. DNA is negatively charged. That's how they could stay stuck together very, very tightly. Chromatin is further compacted into nucleosomes. These are the little beads on a string that make up nucleosomes. So you take your DNA, wrap it around some histone proteins a couple times, you got a nucleosome. So now we're starting the process of condensing our DNA. So rather than just one long naked strand of DNA, now it's wrapped around a histone octamer. 8 histone proteins make up this little apple-looking thing, and so that makes up a nucleosome. Then you have beads on a string, right? So the nucleosomes are beads on a string, and then you could take these, and so they're sticky. Remember, that's your binding agent. So then you could take these beads on a string, start coiling them together. So now you have a coil. And then when you have that coil of a certain filament or of a certain thickness, then you could coil that coil. And that would be supercoiling. And then you could coil the coiled coil. And I know it sounds like I'm just making this stuff up, but literally that's how it works. Once you've coiled the coil, you could take that and then coil the coiled coil. And so I'll stop now. But the point is that's supercoiling. So beads on a string. Way back, we had DNA, like naked DNA. Then we make then we make a nucleosome. Then we have a string of nucleosomes. That's beads on a string. They further get compacted many times over in a process called supercoiling. And so you can see that in the image here. So this is what we started with naked DNA, wrap it around a histone octamer, make a nucleosome. And so then you could see beads on a string, right? So you have those nucleosomes. Take those beads on a string, start wrapping them to make a coil. And so now you have a coil. And so then you have a certain filament of chromatin, a certain nanometer filament of chromatin. And then it just keeps going there. We could describe the different levels by their nanometer filament designation. You have multiple levels, but then eventually you get to the most condensed state. And that would be a condensed chromosome at, say, for example, tail end of prophase. And so you can click this link. It's just a little video showing supercoiling. It's a YouTube video, nothing spectacular. But that is how we fit all that DNA into just the nucleus of our cells.